Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today I will discuss a very important topic with you people, and it is related to bacterial cell wall. And the title of my today's lecture is the synthesis of peptidoglycan. As you know that peptidoglycan layer is very much important for the survival of bacteria. In gram-positive bacteria, it is the outermost layer, and in gram-negative bacteria, it lies after the outer membrane, and it gives strength to bacteria, and it gives support to bacteria, and because of this layer, bacteria can withstand different osmotic pressures and different adverse conditions. Okay, so in the previous video, I have discussed that what is basically the structure of peptidoglycan. And as I have told you that in the next video, I will discuss that how really this peptidoglycan layer is synthesized. So in today's lecture, I will show you that what are the different steps in the biosynthesis of peptidoglycan. So let's start with the video. Okay, first of all, you should know about the gram-positive bacteria and the gram-negative bacteria, that where does this peptidoglycan layer lies in the cell wall. I have discussed it in the previous video, but just to give you a background of peptidoglycan layer, so I have added this slide. Just look to my cursor on the screen. This is the cell wall of gram-positive bacteria, and this is the peptidoglycan layer. Okay, and it is about 45 to 50 sheets. Okay, and it is the outermost layer of gram-positive bacteria. Okay, and if I show you the cell wall of gram-negative bacteria, so this is the peptidoglycan layer. Okay, this is the peptidoglycan layer, and it is thin in diameter. Okay, with respect to gram-positive bacteria, it is about 1 to 5 sheets. Okay, 1 to 5 sheets, and it lies after the outer membrane. Okay, so keep this thing in mind, that in gram-positive bacteria, it is the outermost layer, but in gram-negative bacteria, it lies after the outer membrane. Okay, one thing I want to show you guys is these spaces. Okay, these spaces is known as the periplasmic space. Why I'm showing you, because in the later video, I will discuss about the periplasmic space and its importance. Okay, just keep this in mind, that the space in between the plasma membrane and the peptidoglycan layer is known as periplasmic space. Okay, these are the periplasmic space, and this is the outer membrane, and this is the peptidoglycan layer. So the space in between is known as the periplasmic space, and this is also known as the periplasmic space. So this is a periplasmic space of the gram-negative bacteria, and this is the periplasmic space of the gram-positive bacteria. Okay, so keep this periplasmic space in your mind. I will use this in the later videos. Okay, so this was the peptidoglycan layer in gram-positive and gram-negative bacteria. If I show you the microscopic picture, microscopic image, so in the case of gram-positive cell wall, this is the peptidoglycan layer. Okay, the thick layer. After that, this pink color is the plasma membrane, and this is the electron microscope image. Okay, and if we look here to the gram-negative bacteria, so this is the outer membrane. After that, this dark color is the peptidoglycan layer. After that, this is the periplasmic space, and this last one is the plasma membrane. Okay, so keep this microscopic view in your mind. Now this is the main structure, the molecular structure of a single subunit of peptidoglycan. I have added the slide just because to give you a background. I have discussed in detail the structure of peptidoglycan layer in the previous video, so you can watch that video to increase your knowledge about the peptidoglycan layer structure. Okay, if we take a single unit of peptidoglycan, it is also known as a repeat unit. Okay, so if I take this unit, so this orange color is the N-acetylglucosamine, and this yellow color is the N-acetylmuramic acid. And basically these are glucose molecules, but a little bit modified. Different functional groups are added to these glucose molecules, and these are connected to each other by a glycosidic bond. Okay, after that there are these four rings are amino acids. Okay, these four rings are amino acid that are attached to N-acetylmuramic acid. Okay, so this is the L-alanine, D-glutamic acid, the diaminopamelic acid, and D-alanine. Okay, so keep this structure in your mind. One thing remember in your mind, that in case of gram negative bacteria, this third number amino acid is diaminopamelic acid. Okay, and in case of gram positive bacteria, this is replaced by L-lysine. Okay, it is replaced by L-lysine. So keep this difference in mind. It is important for MCQs, so you can then solve it easily. In gram positive bacteria, it is L-lysine, and in gram negative bacteria, it is diaminopamelic acid. Okay, and this last one is the D-alanine. In the biosynthesis of peptidoglycan layer, to this alanine and another amino acid is added, and it is also known as D-alanine. Okay, here it is not mentioned, but when these subunits are synthesized, so basically it consists of 5 amino acids, but when they are bonded, when they get bonded to each other to this linkage, okay, this bar type linkage, this is known as the pentapeptide or pentaglycine ring. Okay, so when they get attached to each other, so they loses this last amino acid that is known as D-alanine. Okay, so if I erase it like this, so this is and when they get bonded, so they loses their D-alanine. But when they are when they start synthesizing their subunit, so they should have D-alanine as the 5th amino acid in their structure. Okay, so keep this difference in your mind. Okay, now here we come to the biosynthesis of peptidoglycan layer. Okay, first of all, what happens really that N-acetylglucosamine and N-acetylmuramic acid is uh, they cannot bind all alone. Okay, means that they cannot travel inside the cytoplasm of the cell, and they cannot means that they do not know about their pathway, that where should they bind. Okay, so there is a special type of carrier molecule that is known as UDP. Okay, and UDP stands for uridine diphosphate. Okay, it is a type of molecule. It is not a protein, it is a molecule that is known as a carrier molecule, and it carries the N-acetylmuramic acid and tell him that you have to bind to the L-alanine. Okay, and you have to travel inside the cell, and it is a type of carrier protein. If I want to show you the structure of uridine diphosphate, so it is just like this, and this name is bonded here. Okay, then N-acetylmuramic acid is bonded here. So it shows the N-acetylmuramic acid the pathway, that where should it bind and how, and it carries this N-acetylmuramic acid because it cannot travel by its own self. Okay, so it helps the N-acetylmuramic acid in traveling inside the cell and showing him the right pathway. Okay, and N-acetylglucosamine is also binded to it. Okay, it also shows pathway to N-acetylglucosamine, but keep this thing in mind, they cannot bind to each other. They have to bind to uridine diphosphate separately means that a single uridine diphosphate will bind to a single N-acetylmuramic acid or N-acetylglucosamine. Okay, so keep this concept in your mind. Okay, so now if we this was a simple structure of uridine diphosphate. Now we will come here to the main mechanism of that, how peptidoglycan layer is synthesized. Okay, so what happens that uridine diphosphate binds to N-acetylmuramic acid, and it acts as a carrier protein. Okay, the biosynthesis of N-acetylmuramic acid and N-acetylglucosamine. I have not added the slides of that biosynthesis because then you have to get a bit. It should become a bit complicated. Okay, so just for you people, I have not added the slides of the biosynthesis of N-acetylmuramic acid and N-acetylglucosamine. But keep this thing in mind that bacteria can synthesize it inside its cytoplasm. Okay, so keep this concept in your mind. What happens? So now we will start from the beginning. That what really happens that N-acetyl muramic acid binds to this UDP. Okay, and this UDP guides it across the cytoplasm. What happens with the help of different enzymes? First, this L-alanine binds to the N-acetylmuramic acid. After that, the second step is that D-glutamic acid binds. After that, L-lysine binds, then D-alanine binds, and at the last, and another D-alanine binds. Okay, so these all are amino acids, and they binds to each other with after step by step. Okay, first the L-alanine bind, then glutamic acid, then L-lysine, then the D-alanine, and the last one is the D-alanine. So if we count it, so 1, 2, 3, and 4, okay, and 5. So it is known as a pentapeptide. It is known as pentapeptide. Okay, because penta means 5, and 5 amino acids are 5 amino acids are added, so that is why it is known as pentapeptide. And one thing, keep this thing in mind, that in our cell, inside our cell, a ribosome can also synthesize amino acid chains, okay, peptide chains. But here and it is just like the translation process, that in translation process amino acids bind to each other with the help of transfer RNA. Okay, these all are the process, but here neither transfer RNA is involved nor ribosomes are involved. Okay, here are just enzymes that add different amino acids to each other, these amino acids to each other. Okay, so if your examiner asks you that are ribosomes and transfer RNA involved in this process, so your answer will be no. Your answer will be that here simple enzymes are used in the by adding these amino acids. Okay, so keep this thing in your mind. After that, when these amino acids bind to N-acetylmuramic acid and it form a chain, a large subunit. So after that, this UDP has to leave this unit. Okay, this UDP has to leave this unit. Okay, so what happens? This UDP give its 1-phosphate to this pentapeptide chain, this name, and it leave it with the uridine monophosphate. It convert itself into uridine monophosphate and it give 1-phosphate to NAM. Okay, just like this. This is the phosphate of N-acid, this uridine diphosphate. Okay, after that, what really happened? Here come a second carrier. The first carrier was the uridine diphosphate, and this is the second carrier that is known as bactoprenol. Okay, it is also a carrier. And what does this carrier do? This carrier helps the NAM and the pentapeptide to move across the plasma membrane. Okay, keep this thing in mind, because it has to move here. This is a cytoplasm, and it has to move across the plasma membrane. So it has to move across the plasma membrane and enter the periplasmic space just to aid in the synthesis of peptidoglycan. So this bactoprenol is a carrier protein, a carrier molecule, not a protein, that helps this these compound to move across the plasma membrane. Okay, so keep this thing in mind. So what really happens? And this is the main. I will show you the molecular structure of bactoprenol. This bactoprenol is a molecule. It consists of a phosphate. Okay, and after that, it binds to the phosphate of the name, and it form a molecule. And this molecule is known as lipid A. Okay, just follow the steps. Okay, so this is known as lipid A. After that, as I have told you that N-acetylglucosamine will be also carried by the uridine diphosphate. So here also uridine diphosphate carries N-acetylmuramic acid, and here also uridine diphosphate carries N-acetylglucosamine. So it also brings the N-acetylglucosamine, and then this N-acetylglucosamine binds to the NAM. Okay, as I have shown you the structure of the peptidoglycan subunit in the previous slides. So here this is also known, and this is a carrier molecule. So now here by the addition of N-acetylglucosamine, what really happens that this molecule is known as now lipid 2 protein, lipid 2 molecule. Here when N-acetylglucosamine was not binded, so it was known as lipid 1 molecule, and now when by the addition of it, it is known as the lipid 2 molecule. So it is also necessary for the MCQs that you should differentiate between the lipid 1 and lipid 2 molecule. After that, what really happened, as I have mentioned, that bacterial has to pass the this large complex across the plasma membrane. So as it passed this complex across the plasma membrane, directly it leaves this chain. Okay, it leaves this whole compound. Okay, by taking both the phosphate with itself, and this is now already made. It is now a ready-made repeat unit of a peptidoglycan layer. Okay, so this this bacterial leave this molecule, and then goes across the plasma membrane, and reaches its final destination. Okay, and here again it has to pick the UDP-NAM peptidoglycan, this name and peptidoglycan, and it will then again perform its function. Okay, so but one thing, keep this in mind, that this phosphate has to be released. So after that, when it releases this phosphate, so then it can go across the plasma membrane. What happens with this unit? Then after that, this name and neg and this pentapeptide ring attaches with the remaining subunit of the peptidoglycan. Okay, so these were the remaining these were the already existing peptidoglycan subunits, and then this when this subunit adds with this peptidoglycan layer. Okay, so and it increases its length, and different other things happens. Okay, so just follow the steps from 1 to 8. After that, when they binds to the peptidoglycan layer, and this is basically the glycosidic bond. After that, here is the last step is the peptide cross link between peptidoglycan. It is also known as transpeptidation. Okay, what is transpeptidation, and what is what basically this is, I have discussed it in the previous videos. Okay, I will also throw light on this transpeptidation a little bit in the later slides. Okay, so up till now, they have added to the peptidoglycan layer. Now what they should do? They have to make interbridge in between the peptidoglycan. Okay, so one thing, keep this in mind, this is the bacterial pyrophosphate. Okay, as I have told you that I will show you the molecular structure of bacterial. It consists of 55 carbons. Okay, if you count it, so it is 55, and to this the name can bind. Here name can also bind, and neg can also bind, but keep this in mind, they have to bind separately, not combinedly. Okay, so either neg will bind or name will bind. Okay, it depends on the situation. So this is the structure. Just you can use this slide in your assignments and presentations. Okay, now this is a transpeptidation reaction. This is the after the addition of the subunit of the to the new subunit with the existing peptidoglycan layer, it has to make interbridge in between the two molecules, just to give strength to the peptidoglycan layer. If I show you here, okay, so this is the pentaglycine interbridge. It is also known as peptide interbridge. Okay, so it has to make interbridge in between these layers. Just why they are making it? To stabilize the peptidoglycan layer and to give strength to the peptidoglycan layer. Okay, so how this occur? Okay, this one. So after that, the last step is that it has to perform transpeptidation. So in case of gram negative bacteria, this is the gram negative bacteria and this is the gram positive bacteria. So there is a little bit difference. What really happens that D-alanine, the COOH of D-alanine, what really happens? The COOH of D-alanine bonds to the NH2 of the diamino pamelic acid and it loses this D-alanine. Okay, here also the D-alanine is lost. Okay, as I have told you that after when they get binded, so they have to lose their D-alanine at last. Okay, so here is a peptide bond. Okay, here is a peptide bond and after that they get binded. So this is the peptide interbridge and this process is known as transpeptidation and this is the peptide bond. Okay, if I write it PB, okay, so this is the peptide bond and it occurs inside the cell wall of the gram negative bacteria. What really happens inside the gram positive bacteria that five amino acids and those amino acids are glycine. What they do, they come and form a pentaglycine interbridge. Here the here what happens, just the two amino acid binds to each other through a single peptide bond, but here five peptide bonds are formed because five amino acids come here and binds to the bind the two subunits together. Okay, so this is the NH2 of the and this is the NH2 of the glycine and here this is here will be the COOH of the here will be the COOH of the D-alanine and this L-lysine here will be the NH2 of the L-lysine. Okay, and it will get binds to the COOH of the glycine. Okay, but here will be five glycines. Okay, that's why it is known as pentaglycine interbridge. Okay, so this was the final step of the peptidoglycan biosynthesis. After performing these steps, what happens that it completes the biosynthesis of the peptidoglycan layer. Okay, so hope you have understand about the biosynthesis of the peptidoglycan layer. The last slide is a bit informative because most of the student ask that when the cell is dividing or at which time the cell wall synthesis starts. Okay, and where does it starts and what is the actual position of the starting of the peptidoglycan layer synthesis. Okay, so I have added this slide. So in case of gram positive bacteria, most of the coccoid shaped gram positive bacteria, what really happens that when they start their binary fission. Okay, when they start their binary fission and so at the last when there is septal formation, when the cell starts to divide and here is a groove like structure and it starts to divide, so at that time the peptidoglycan layer starts. So the synthesis of peptidoglycan layer starts at this point. Okay, here also and here also. So just like this, it starts, this blue color is now the peptidoglycan layer is synthesizing. Okay, so after that when it's sent, so half of the peptidoglycan layer, these are the new one. This was the old one. Okay, this was the old one and this is the new one. Okay, so half new peptidoglycan layer is received by one cell and half is received by the other cell. So your answer will be to your examiner that when it starts in the gram positive bacteria, when the groove starts formation, so at that time the peptidoglycan layer synthesis starts. Okay, and what happens, and this is the position. Okay, it does not start here or here or here, it starts at that position where the groove starts its formation. Okay, so keep this thing in your mind. But in case of gram negative bacteria and when it is especially bacilli bacteria, so when it starts dividing, so at different position the peptidoglycan layer can start its formation. It can for example, this is the middle region. Okay, so it can also start its biosynthesis here, but it can also start synthesis here and here and here. Okay, so Now you have become clear to this question, that in case of gram positive cocci bacteria, only the peptidoglycan layer starts its synthesis at the septal region. But in bacilli bacteria, and especially gram negative bacteria, it can start its biosynthesis at different positions. Okay, and after that, when it gets divided, so half of the peptidoglycan layer will be new and half will be old. Okay, so this was a little bit informative slide. I want to share it with you people. Okay, so hope you have understand about the mechanism of the peptidoglycan layer, that how it is synthesized. If you have any question related to bacterial cell wall and synthesis of peptidoglycan layer, so you can ask me in the comment box. I will answer you there. Okay, and in the next video, I will discuss the action of different antibiotics in the destruction of the peptidoglycan layer. Because the antibiotics that are the cell wall inhibitors mainly stops the different reactions. Okay, mainly stops the different steps involved in the peptidoglycan layer. So in the next video, I will discuss that how antibiotics or the cell wall inhibitors destroy the cell wall of the cell wall of bacteria, and how it stops the different steps of the peptidoglycan layer synthesis. Okay, so thank you for watching this video, and thank you very much. God bless you.